with genera-wise equally or almost equal distribution in the neotropics as well as in the neotropics, but species-wise, more distribution was the Old World region. Around two-thirds of this distribution is found in the Old World, while one-third is distributed in the neotropics, with a few occurring in the Europe as well as in Southern Asia. And inside, the family is widely distributed by 24 genera and 150 species. If we can see that the genera of the family includes 41 Kengilia 41 species, followed by Didymocarpus with 28 species and Eskinanthus have 20 species, while most genera are represented either with one species, two species, or three species like that. So altogether, the family is represented by 24 genera and 150 species. This is the classification of the family Gesneriaceae, and we have seen on the left side you can see there's a classification given by Smith in 1996, in which he recognized only two subfamilies, while Weber et al. Recognized three subfamilies. The subfamily Anangoideae, which is monotypic, mostly distributed in the South America, while subfamily Gesnerioides is the neotropical Gesnerioids, which include a few Old World members like Titanotrichae as well as the hemispherical Ornithoroideae. While the subfamily Didymocarpoideae, that's actually the Citandroideae of the Old World, is represented by two tribes Epithymatae as well as Trichosporiae. And the two tribes are divided further into 14 subtribes, while three subtribes of the tribe Epithymatae is represented in India, while 6 out of 10 subtribes of the Didymocarpoideae. So in total, it is represented by 9 tribes in India. This is overall of the morphological features of family Gesnerioideae. We can see it is they include mostly perennial herbs or rather soft stem shrubs. So with regard to the habit diversity, it includes members, plants like herbs, shrubs, or even climbers and other climbers, etc. While the leaves are generally opposite, with sometimes they are occurring in birds, especially towards the apical region. The flowers are pentamerous, consisting of bisexual flowers with zygomorphic corolla. While most interesting regarding this Gesneriaceae are the pair flowered cymes. Usually we know in the cymos inflorescence, the flower ends singly. That means the ultimate unit of cymes ends in one flower. But in Gesnerioids, we can see an additional flower. That means it is called as the front flower, is formed along with the ultimate cyme unit. So in all cymos segments or cymol units of the Gesneriaceae, we can see instead of the one flower, we can see two flowers. That's the most unique character of the Gesneriaceae. And this character is also in some families like Acidaceae as well as of the Loganiaceae of the Lamiates as per the APG classification. And this ovary, again, we can another character is a parietal placenta with numerous ovules. So this is the research we have undertaken so far at Calicut University. So far we are working on this plant group since 2013. We have undertaken the revision of this family in southern India, as well as also undertaken an integrative approach involving the molecular, cytology, micromorphology, anatomy, pollination, biology, as well as ontogeny of different plant species. And so far we have described several species in the Engidia, as well as few species in Dysphenotus and few species in Eskinanthus, and few new records, new typifications, etc. And this is the external funding we received as well as for the collaboration. And we can see, I myself visited Wadley in the Royal Botanic Garden in Peru, and also at the Institute of Botany, Chinese Academy of Sciences as from fellowship from the Indian National Science Academy. And two of my students are also collaborating or work in the Royal Botanic Garden, Edinburgh. So we received the funding from Sibal Fellowship from the Royal Botanic Garden, then Just Area Society's funding through Elvin McDonald Fund. IAAT research grant also awarded to my few of my students. And currently, one student is working as a postdoc fellow with the DST Women Scientist Scheme. So I am also happy to share some insights on the few selected genera of Gesnerias, especially of Jardonia. We know this is a monotypic genus and represented and is endemic to the southern western Ghats. We know the distribution is limited only to few locations in the southern western Ghats, and this plant is quite unusual among the Gesneriaceae because of the isopotalus. It's usually for Gesneriaceae we can find the anisopotalus one. So here it is an isopotalus, and the Opodospermus seeds is again another character of this interesting feature of this Jardonia. Then another one you can see on the right, in the left side on the right, you can see there is the flattened appendages on the filaments. I don't know. So we are working on what is the significance of such flattened filaments, especially with regard to the pollination mechanisms, other things. So there are appendage filaments there, and again instead of the unilocular or the usual character of the Gesneriaceae, we can find four separate classes in the Jardonia. And most of the other classes we kind of put this Jardonia in the Jardonia or some other families, but we are working on because of the fair flower cymes as well as parietal placentation. It is retained in the Jardonia, and also in the molecular phylogenetic analysis we can find the position of Jardonia under the basal genera, where they find it is treated in a monotypic subtype Jardonia. So my student is Ms. Vahida is presently working on the floral ontogeny as well as the other mechanisms of this particular species to give more insight on the mechanism of the reproduction also. Now this is another one plant we worked on earlier on the Ringoglossum, Genisha what? And the genus is represented by 5 species in India, and we have in South India 3 species, while 2 in the Northeast India. At the same time, we are also collaborated with Subani in the Sri Lanka and also with Michael Muller in RBG. So ultimately, we have developed an integrative approach in which the samples from both Sri Lanka as well as South India has been, the data from both sides have been integrated, and we ultimately produced an integrated revision of Ringoglossum for South India as well as Sri Lanka, in which what's more interesting is the one particular species has been reinstated from the synonymy. So you can see there is 2 pictures on the right, on the left side, 2 flowers were given, 2 photographs were given, in which on the top you can see both looks like blue, but at the center we can see there's an yellow patch at the center, while on the second flower we can see it is completely blue. So it's only morphological indication that it may be separate, but in fact Dalzell has described the below flower as a separate species called Ringoglossum scabrum, while the other one is described as Ringoglossum nocturnum, but long been Ringoglossum scabrum has been placed under the synonymy of Ringoglossum nocturnum. So we have sampled the materials from both sides, and this is a classic example where how the Sri Lankan, if we integrate or we collaborate with the scientists in the Sri Lanka as well as India, because they have lot of similarities in the distribution, and also there is a lot of endemism also. This classical example where we can see the data can ultimately produce more insight on the resolution of this, because it is also sitting here. That's right. So ultimately so we have data from the morphology, molecular as well as the micromorphological side. Ultimately we reinstated Ringoglossum scabrum as a distinct species and published in Plant Systematics and Evolution. This is another interesting genus, this Tengilia, but I am focusing on a particular species called Tengilia prodiviana, which we described in reading in 2012. And the plants were initially collected by Dr. Pradeep as part of his research project, but he collected with the wood flowers as well as seeds, so he could not identify properly where it belongs as. So, and almost 10 years later, when Dr. Manudev visited the same or adjoining localities, he collected this interesting plant, and upon further investigation, we found that this species is quite interesting, especially with respect to the flattened tubers. Now, no, so regarding the Kengelia, we know the Kengelia is distributed. There is a habit-wise separation, especially the coalisant species are distributed in the Sri Lanka as well as the northern India, while coalisant species are confined to South India, except one species which is also occurring in the Sri Lanka. So we have made a detailed studies, student is working on that. But what is curious on this particular plant is the flattened tubers. You can see the flattened tubers there. This is not represented in any of the South Indian species. Even the 14 up to the 15 species occurring in South India are endemic, and none of these species have this type of flattened tubers. And another one is the peculiar fruits. They can see the fruit, which is usually for all Kengelia, the fruit elongate once, but here you can see the globose fruits which decays on the upper side. So these unusual characteristics, ultimately we have, my student Janisha is working on that. She has made a phylogenetic analysis of that one, and ultimately found that it's also from a separate clade in the molecular analysis. So very interesting discovery on a particular species in the morphological side, and we are again working on in what way it is have some evolutionary significance. So let us, I know you are all hungry, and the time exceeded. I don't know. I may skip some slide on this is my major topic. So this already what happened. This is only an introduction regarding the what we have undertaken in my lab on the Gesneriaceae. In between, I may skip some slides. I know we are all in a difficult. sorry for. Now let us see. This was actually Eskinanthus, this is very interesting genus, and one of my students, Mr. Akhil, is working on that. He also received, he's the middle person, he's worked on the Eskinanthus of India. We have a program by the Sciences of CRB. The CRB is funding this program. In meanwhile, he also received the IAAT research grant. He also got the fellowship from the RBGC, but fellowship he got. Then also he got funding from the Gesneriaceae Society under the Elvin McDonald's Endowment Fund. So ultimately, he started his work in 2018. He enormously or extensively explored the various parts of South India, the entire India, because this plant is the genus is exclusively distributed in the northern Indian region. The Indian level is considered. We have only one species in South India. So just see the general features of the plant group, include the epiphytic or lithophytic curves, and what the predominantly or exclusively bed-pollinated habit. Then the protandous flowers, and mostly the flowers are scarlet. Very rarely you can see green or white corolla, and there's again coherent in pairs. And the most striking character of Eskinanthus is the long, narrow capsule and the appendage of the seed. So the seed is appendage at both ends. On the hilar appendage there is one, apical appendage there is one trichome, while on the other end there is one to many are present. And this plant is usually called as the lipstick plant or brushworts. And in the Indian side we have around 20 species. Worldwide it is 181 is represented with 11% of distribution in the peninsula in the Indian zone. So the genus was initially described by Dawn under the name Trichosporum with two species, Trichosporum grandiform and Trichosporum parviflorum, based on collections from village Nepal. So trichos, in fact, the name was derived from the hair at stage. And while William Jack, again, it should be an inspiration for the youngsters. He described that particular genus at the age of 27. Maybe none of this student, I don't know, is sitting with that age group, but Jack described, the total lifespan is 95 to 22, it's only 27. And he passed away, lost his life at that time, but established this genus Eskinanthus with two species from the Sumatra, with Esquinanthus volubilis as well as Esquinanthus radicans. And even though so we know as per the rule of priority, the trichospora has priority over the other one, but during the years more species were described under Esquinanthus than the trichosporum, and ultimately, based on the proposal from Sprague in 1929, it has been accepted as a conserved name in 1935. So beautiful illustrations of this particular group can be seen in many publications, especially this Mary Mendel. This is the latest work, even though there are contributions by Bentham, they are classified as Esquinanthus, Bentham, then Clark, all made classifications of this particular group, but Mary Mendel has made the extensive survey. So she in fact intended to publish a monograph of the Esquinanthus in the world. But unfortunate of his sudden death in 2004, this project was not being completed, but she is a good illustrator. We can see the beauty of the plant she portrayed there. She's actually from the, she's from Kew, and a lot of publications, around 50 species has been published. And you can see the number of list of publications in 95, 98, 1990, so a lot of publications into her credit. And this is illustration by drawing by her. And on the right side you can see another image, another illustration by Middleton on the species named after Dr. Mendel, Esquinanthus benthumii. So these are some of the beautiful illustrations we can see on various books, maculators by Lindley, and another one of by Hooker and Thompson, and the last one of the Roxburgh icon. See, this is regarding the what all works already have been undertaken in different areas. You know, the genus is principally Asiatic, the genus is principally Asia, Southeast Asian. With you can see the colored area where the genus is principally distributed. But during the past few years, the genus has been studied. China by Wang et al. 1998, Myanmar by Kress et al. 2003, Thailand by Middleton also revised Cambodia, Laos, Vietnam, Singapore and Peninsular Malaysia, and Sulawesi by Bentham and Atkins in 2003, while it's very difficult to pronounce the last one, it is in Sumatra, it's in 2009. So for India, Bhattacharya and Goyal made a revision in 2015, but in fact, in 2000, a few years later, I forget the exact year in which one other person has published the Northeast Indian revision, in which they recognized only 16 species from the Northeast. So this is was undertaken by Middleton as the recent of the contemporary scientists who have enormously worked on the Esquinanthus, in which my student also have discussion with him. And this is the molecular work already available on this particular gene, it's difficult to pronounce, and Chrome performed a phylogenetic analysis of the genus using 8-year sequence. And 50 species of Esquinanthus were selected as in group. And anyway, there are no samples from the Indian side, but few samples of species that occur in the neighboring regions. But generally speaking, few species are, majority species from no species from the Indian side are included. And this is the distribution of the Esquinanthus in India. So this Esquinanthus in the world level, we can see the Southeast Asian region with a lot of species, and there we can find Indonesia having the maximum diversity with 45 species, followed by 32 in the Philippines, 31 in China, and so like that. Thailand 22. So this is concentrated mostly in the Southeast Asian countries. So this is regarding I will quickly pass through the few slides. So this is the diversity we can see with respect to the habitat. It is usually found as on the trees mostly. And again also, my student also found a lot of difficulty, especially for collecting, because they are usually adapted to live on the higher branches, in the top branches only, not on the lower sides. You may be knowing we are all exploring these Northeast Indian regions. So where it is very difficult to get collecting the plant materials, but if the conditions are ideal, they even grow on the rocky substrata as the lithophyte also. So that was the habitat diversity. You can see on the left image, the plants are growing in high canopy. Even I myself also visited several times in the Northeast. Very difficult to collect these plants of Esquinandus. This is the morphological characteristic of this particular group, general morphology. We can see what is most diagnostic are their peculiar fruits, especially the long, narrow capsule used. And you see the beauty of the flower on the left side, the corolla, calyx, bracteole, and peduncle. And the leaves, they are basically opposite in the phyllotaxy, and usually they are very rarely they are ternate or whorled towards the apical region. In few genera, not few species occurring in the adjoining countries, they are in the ternate or whorled phyllotaxy. And one other character is also the thick and leathery leaves. The plants are exposed to higher temperatures, so they have their own adaptation with thick leathery leaves. And sometimes in the one particular group, especially for now the scoring group, where we can see marble leaves on the right top. Then inflorescence, as I said, the basic pattern of the inflorescence for a Gesneriaceae is pair-flowered cyme, but unfortunately that pair-flowered cymes are greatly reduced here, but the inflorescence pattern will be either axillary, one to many flowered cymes in the axillary zone, or it may be pseudo-terminal clusters. So the calyx, very interesting feature for the identification of the individual species. Their sepals are 5 free to the base, or sometimes may be variously fused. So this is a particular character for the identification of the species at the species level. And again, in some species, they form the tubular calyx. So, and this tubular calyx sometimes collect a lot of water and helping to keep away the herbivores. So this is an adaptation they developed during the course of evolution. That's what is called as the water calyx. This type of water calyx is very rare in other genera or other groups also. On the extreme right, you can see the water calyx. On the extreme right, you can see. So there are much variation in the nature of the calyx, from the completely free to the slightly fused to completely fused. So in that pattern, you can see the calyx. Then corolla is the most beautiful part of this Esquinandus, where you can see, as I said, that they are zygomorphic, tubular, and curved. So the curved nature of the corolla, and again comparatively very long corolla, and it is adapted for the strongly zygomorphic one, protandus one, and greatly adapted for the bed pollination. And also they are for attracting the pollinators. A lot of nectar is also produced at the base, and the corolla. The top region is divided into 5 lobes, and 2 upper lobes, 2 lateral lobes, and a lower lobe. And again, what is striking is very difficult to define. Even my student also found it very difficult to define the corolla, because lot of markings on the inside of the corolla, and even the curving of the corolla is very difficult to define, and even the nervation is so very difficult to define. Even though you feel it's very simple, so in the inside there are a lot of markings and everything. How to describe in a corolla is usually very interesting and difficult. So you can see the variations in the corolla there. And next one, it is anthecium. It is again 4 stamens are there in 2 pairs with a 1 staminode. And in very rarely the 4 stamens are fused, as in the case of chiritoids. On the right side you can see how all the filaments remains free. All the filaments are free, but at the anthers of all the stamens are fused in the case of chiritoids. While for other genera, so other species, you can see they are 2+2 pattern. So the two anthers of the frontal stamens are fused, while the lower 2 are fused. So the 2+2 or the fused in the top level. That's the case of chiritoids. Now let us see, look on the gynoecium. This is still superior composed of 2 carpels. And again, this is a very interesting feature. We can see at the base of the gynoecium there is a 5-lobed annular or 5-crenate type of disc, and below the ovary there is a stylus present that is also retained in the fruit also, ovary and ovary and style are glabrous, or with sessile glands or pubescence, etc. And the fruits, as I said, is very diagnostic for its identification at the species level, and they are low and narrow and locally seedal. The seedless and the sterile portion of the ovary is developed into a seedless portion at the base, corresponding to the site of the gynoecium. And again, the individual seed consists of three parts. The grain is there and appendage around the apical side. The apical appendage means that's what's seen on the hanging side is called as the apical appendage. One that is attaching on the fruit is called as the hilar appendage. And here, the number of hilar appendage, the nature of the ornamentation of the seed grain as well as the apical appendage are very diagnostic, especially at the classification of the genus, the sectional level, etc. So this is a sectional classification. Because of time limit, I am not going further, but I can see the same characters are the primary characters that are used by the earlier authors to classify. Bandham was classified into few sections. Clark, then Wang has classified. Mendham also has classified. But see according to the number, number of the seed appendage, hilar appendage, whether it is 1, 5, or 2 to 4. That's the primary character used by them. Along with some other characters, mostly the number of appendages are the primary criterion for the classification. This is the classification. You can see at the word level, you can see there are 6 sections are recognized, but for India we have representations for 4 sections. And this is the some micromorphological studies we have undertaken. We have sampled 6 or 7 species belong to different sections of the Echinanthus in India, and our studies with molecular data is not supporting the sectional classification revealed by other groups. And this is the palynological studies we have undertaken. We again sampled few representing different sections. Because of time limit, I am just passing on to quickly I'm going. And what is interestingly we found is the presence of the orbicules in the pollen grains that are not even reported to Botanical Survey of India directed. This is Manthus, Botanical Survey of India directed. This is Manthus. So this is parasitic, one of the widely distributed species in the Indian context. We can see this is widely distributed and also exhibit in the Indian context. We can see this is widely distributed and also exhibiting variations at the that some of the variations, the leaf size can be seen, at the calyx level can be seen, and floral level can be seen. This is again another interesting species, also widely distributed, Parviflorus, also exhibiting variations. Now there's again Perotacti. This is the only species distributed in the southern Indian regions, and lot of discussions needed on the first particular species, but I in zone, but we have the molecular study does not support such the variations represent South India, maybe the South India as well as Sri Lankan species may be united at a later stage by one species. This is another species we described from Manipur. There are a lot of stories to explain about this interesting discovery. We ultimately we discovered this particular plant from that area. The other person also collected this same species and both communicated to the same journal. Interesting one. So interestingly, the material received for me for evaluation from the other side. So ultimately we decided to combine the article instead of going for separate publications. So that's why that's happening in that way. So this is again a genus Echinanthus. Echinanthus superbus is the most beautiful plant among Indian region. We know there is. I told at the beginning that the flowers varying from mostly dark in color, but there are variations from white in some species, white, some species maybe. In outside India, we can find a lot of color variations, especially with respect to green, yellow, etc. Even Mendham has described yellow species there. Then Tirapensis, Volubilis. So an overview of these studies they undertake 20 species in foreign endemics. 90% of distribution in the northeastern region. The sexual classification based on the seed morphology has no phylogenetic support. Pollen grains are generally Stenopilinus and presence of orbicules reported. 2 new species and 3 variations, and this is the overall beauty of this particular group. Even anyone can try this particular plant for their homestead gardens. See, this is all from the net, only not from my experience, but they are taken from the net. So it is very suitable for growing in the hanging baskets. And again, quickly through few slides I can go. The other work we have undertaken in our laboratory. We are also working on the utricularia. It's a very interesting group. And we know at the Indian side, Dr. Jana has made an extensive survey and documented in 1992. But we are now focusing on the utricularias of India with molecular data that the revision of the molecular-based, morphology-based taxonomic revision. And two other students are also, they are sitting there. Vaishnavi is working on the trap architecture, feeding structure, as well as on the ecological preferences of selected species of this electrical area, because most of this endemic and their limited distribution in the northern plateau of the plateau of the lateritic plateau of the Kerala as well as in the northern western Ghats. And one other student, Mr. Krishna Priya, is working on the molecular taxonomy revision, while one more student, Sai, is working on the trap architecture that has been supported by the Science Engineering Research Board. So through SEM as well as GEM study, we are going to explore the dimensions in the trap architecture. This another one that we have also documented the diversity of Idukki district, Kerala. We have undertaken this is one of the largest districts in India, and a lot of discoveries, especially with respect to the new species. Around 16 new species has been described, and this program has been supported. 3 students have worked on the group, and we documented. So the student also receives several awards, Mandala Award, so a lot of awards also the students got. And it's very interesting from the group to work on the Idukki district. This is another one. We have worked on the zonal of the Velastomataceae, around 49 species and 1 variety, and we are also working with Dr. Reshmi on that particular group. We have done, we also received the funding from the International Association for Black Taxonomy. Then Aspire Research Grant she received, the Royal Botanic Garden also to support some part of this study, and a lot of typifications, reinstatements, synonymization, etc. A lot of parts have been done by Dr. Reshmi. And another one, so this is the one group that we have extensively studied, especially I started working on the chromatography in 1998 when I met Dr. Robert Fred and Q at Royal Botanic Garden. So he inspired me to start a work on this chromatography, to start there. And 3 4 students have already completed their PhD. One writes the taxonomy, the Comelina, then another on the 2 students completed the revision on the Mordania, and one student on the molecular side, one student on the morphological side, one other student worked on the cytometries. So these are the summary of now one student, and again one student have the pollination biology of selected species. A very interesting group to study the pollination biology also, and the cytology of the endemic Mordania has also been undertaken. And this is Gentinis, this is one work we have studied at the earlier time, so where the new genus called Canscorinella and one species, new species, and all the major genera has been revised at the South Indian level, and a lot of typifications, reinstatement, etc. On the right side, one of my students is Rajishma sitting there, is working on the Ixora on the Indian side. Then another one very interesting study we have undertaken with support from the SERB is the revision of the Idesima. Dr. Manu is sitting here. Manu done the morphology-based revision at an initial time, and the second guy has done, and I don't has done the revision on the support based on the molecular data.
Dr.
So
Nambi,
this is the finish. Finish. So
thank you, Dr. Nambi.
This is my entire group of very enthusiastic team I have at Calicut University. Thank you, sir. Thank you for your patient hearing.
Thank you, Dr. Nambi. It's a very hardcore subject. I think youngsters can discuss with him so that he can guide and give a new way of doing research. Due to want of time, now I pass on to my co-chair to wrap up this program.
Thank you, sir, for this very informative talk. Your work can be an inspiration for the budding and young science taxonomists. Sir, now, we would like to felicitate.
Thank you, sir, for your talk, which was very enlightening. So I request now the chairperson, sir, and co-chairperson, madam, to felicitate Dr. Santosh Nambi, sir, on behalf of Assam University, Silchar. Thank you, sir. Ma'am. Before we come to the end of this Dr. Devajiti Bhattacharjee sir to make an important announcement. He will be presenting the certificates to the co-chairperson sir and co-chair madam sir. Sir, with permission from chairperson sir and Madam shall we end this session? Thank you so much sir. I thank everyone for attending this session. We will now take a break for lunch and assemble for the second technical session post lunch. Thank you all.
Lunch is ready outside the hall. You please have your lunch.